It's a great privilege and honor for me to introduce our keynote speaker for today, which is Dr. Nim Tottenham. Dr. Tottenham is a professor in the Department of Psychology at Columbia University, where she runs the Developmental Affective Neuroscience Lab. Her lab focuses on the development of neural circuitry underlying child and adolescent affect, the external manifestations of people's experience of emotion. Her lab is particularly focused on the neural wiring between the limbic system, which mediates emotion, and the cortical regions, which are involved in higher order cognitive processing. This research focus is framed in the context of early childhood environments and how the ways in which children are raised affect their neural connections and thus later emotional development and behavioral outputs. Dr. Tottenham received her bachelor's degree from Barnard College of Columbia University in 1996, where she was a psychology major. She then went on to the University of Minnesota for her PhD in child psychology and neuroscience under her mentors at Minnesota, Dr. Megan Gunner and Dr. Charles Nelson, and her Sackler Institute mentor, Dr. B.J. Casey. Dr. Tottenham's doctoral work, focused on the development of facial recognition and facial expression processing. In 2006, she began a short postdoctoral fellowship with Dr. Casey at Weill Cornell Medical College in New York City. Soon thereafter, in 2008, she joined the faculty at UCLA, where she remained for 6 years before returning to New York and Columbia, where she is now a full professor of psychology. Dr. Tottenham's lab approaches the questions of early childhood environment effects on neural development and subsequent affective behaviors from a couple of angles. One is an imaging angle, where the lab uses techniques such as fMRI to understand and identify key sensitive periods for environmental effects on the normal development of appropriate neural circuitry. Her lab also seeks to understand the effects of early childhood stress on the development of the brain. Over the last decade and more, Dr. Tottenham's research has been well funded by the National Institutes of Mental Health, among many, many other funding sources. Her work has been widely recognized with a variety of awards, including the American Psychological Association's Distinguished Scientific Award for Early Career Contribution to Psychology in 2015, and the Developmental Science Early Career Researcher Prize in 2010. She and her work have been featured in a variety of print news outlets, including the Washington Post and the LA Times, and television and radio media such as NBC Nightly News and NPR. Dr. Tottenham has been active in teaching at both the graduate and undergraduate levels and earned UCLA Psychology's Distinguished Teaching Award in 2013. She also possesses a very impressive service record that ranges from being a peer reviewer for submitted manuscripts at more than 30 different scientific journals to serving the community, including as a board member for the Adoption Foster Care Therapist Network. In her career, Dr. Tottenham has been invited to deliver over 100 talks. And I think you'll see why. She's a clear, dynamic lecturer speaking on topics of remarkable relevance and interest. Today, she'll speak to us about emotional brain development and the role of early experiences. Please join me in welcoming Dr. Tottenham.
Good morning. Thank you so much for the invitation to speak here today. I'm very excited to tell you about the research that we've been doing in our lab. So the question that we focus on is a fundamental question in psychology, which is how do we explain this enduring link between early experiences we have, whether they're good experiences or bad experiences, and later emotional functioning in adulthood. And so in particular, we're very interested in our ability to regulate our emotions. Now, humans regulate our emotions better than any other species on the planet, but it takes a long time for us to get there during our development. And we focus on brain development to help us understand the glue that links these two experiences and later emotional functioning. And although emotion regulation relies on many, many different regions of the brain, today I'm going to be focusing on the connections between the amygdala and the medial prefrontal cortex. Now, as most of you are aware, the amygdala is an old structure. It develops fairly early in the human, and it's responsible for a lot of our emotional attention and emotional learning about the relative safety and danger of the environment. And in adulthood, the amygdala has strong bidirectional connections with the medial prefrontal cortex, which is really well positioned to gather information from many regions of the brain, synthesize it, and that serves a large purpose of helping to quiet the activity of the amygdala and prevent over-arousal. And as I'm going to explain to you today, that connection or that bridge takes a very long time to develop in humans. And we studied the development of this system during development to really try and ask the question of what is it about the early developing brain that makes it so amenable to environmental influences. Now, if we look at a number of mental illnesses that are associated with difficulty in emotion regulation, they tend to show their peak age of onset around the end of childhood, beginning of early adolescence. And so that's gotten many people excited about looking at this early adolescent period. But our approach, since we're interested in the role of the environment, is to not focus on these red areas, but to study the antecedent period, to try and understand what was happening during development that then led to these individual differences that we see emerge during adolescence. And so if we look at what do we know about human amygdala development, we can start with structural data, which is really just telling us about the volume of these regions. And we know that the basic neuroanatomical architecture of the amygdala is present at birth in the human. And then between birth and your first birthday, we see this massive growth in amygdala size. And we get a little bit of change after that and another blip around puberty. But all of those subsequent changes are dwarfed by this change that we see, this massive growth in the first year of life. And so the question that our lab asks is, what are the functional changes of the amygdala that rest upon this early structural development? And so there are many ways we can approach this question, and one of the ways we've done this is to show participants pictures of fearful faces. Now, there are a lot of pros and cons of using this stimulus, but the reason we chose it is that we wanted a stimulus that would activate amygdala circuitry that we can use in children as young as 4 years old. So we can't use electric shock in 4-year-old children because parents are reluctant to bring their kids in for those studies. So we invite, this is a cross-sectional study here. Participants are between the age of 4 and 22 years old. We put everyone in an MRI scanner. We show them these fearful faces, take pictures of the brain, take all of the brains, smush them together, and we look to see what regions of the brain are changing as a function of age in response to these fearful faces. And the right amygdala emerges, and right is left and left is right in these images. What I'm showing you here is a negative correlation between age and amygdala reactivity to those fear faces. So I extract the data here to show you this is age on the x-axis. This is amygdala reactivity or bold signal in the y-axis. And what you can see right away is that this is the negative correlation I was describing. The younger we go, each dot here is a participant. The younger we go, the stronger the amygdala signal is to those fearful faces. And that effect has been replicated across many labs now using other types of stimuli. So this is not a function of the stimuli that we're using here. So what that means is the average 5-year-old has a much stronger amygdala response than the average 22-year-old. And that makes a lot of sense because the amygdala's primary goal is to learn about the relative safety and danger of the environment. And when you're young and you're new to this planet, you have a lot of learning to do. So you want the amygdala up and running at an early age. But the next question we can ask with these data are, what is the developmental trajectory of the connections between this amygdala response and prefrontal cortex? So to do that, we can use a functional connectivity technique, which if you're not familiar with it, what that means is you extract the time course from one region like the amygdala, you plot it, you extract the time course of prefrontal cortex, for example, and the degree to which they're correlated with each other gives us an index of functional connectivity. In essence, it's suggesting that these two regions are talking to each other. In healthy adults, what you often find with the amygdala and prefrontal cortex is that they're inversely correlated with each other. So when one region, like the prefrontal cortex, increases its activity, the other region, the amygdala, decreases its activity. And so that pattern is very consistent with a regulatory relationship or an inhibitory relationship between these two regions, which has been verified with subsequent lesion techniques. So to demonstrate how this works, if you imagine that each dot here is a person, and on the x-axis is that connectivity measure, the more negative that connectivity, the more inversely correlated they are, the more the amygdala calms down over the course of the session for the individual, those individuals tend to be lower in trait anxiety and show lower autonomic arousal during stressors. So the punchline is, in healthy, emotionally regulated adults, we want to look for that anticorrelated pattern between amygdala and prefrontal cortex. So now what do we find across development? So we take those same participants, and now what I'm showing you here is not activity, but that connectivity measure between the amygdala, which you can't see, and the prefrontal cortex. And we again see a negative correlation with age. So I'll pull out those data and to help us understand that better. So here's the connectivity pattern in our young adults between amygdala and prefrontal cortex, replicating what I just described, where when one region increases its activity, the other decreases. In younger and older adolescents, we still see evidence of that negative connectivity pattern, but what you can appreciate is that the magnitude is significantly decreased relative to adults, which is consistent with what we know about the late development of emotion regulation abilities, even throughout adolescence. But what we were most interested in is what we observed in children, where we no longer see that anti-correlated pattern, but now we get a positive correlation between amygdala and prefrontal cortex, which suggests at the very least that children are not showing this regulatory pattern between amygdala and prefrontal cortex, and there is this shift in the way that amygdala and prefrontal cortex are talking to each other that occurs between childhood and adolescence. We've also found that that shift in brain connectivity is associated with changes in normative developmental anxiety. So for example, young children are more likely to exhibit normative separation anxiety, and then that disappears as they enter adolescence, and that's tracked by these changes in connectivity patterns. So to show you in cartoon format what I'm describing is, in response to fear faces in the adult state, when prefrontal cortex increases its activity, amygdala decreases, suggesting an inhibitory relationship. But in the child state, we do not see that pattern. They're not showing evidence of this regulated pattern independently. So putting those two types of data together, if this is age on the x-axis here, we see that at least in childhood, we get this really robust amygdala response. But it's happening in the absence of this adult-like connectivity between amygdala and prefrontal cortex, which we don't see emerging until the beginning of adolescence. So that's made our group very interested in this childhood period as a potential candidate for identifying sensitive periods of development for the circuitry when the environment may have a particularly profound effect on structure and function that we'll see later on in maturity. Moreover, as I'll explain, we are positing that this strong amygdala reactivity is actually a biological prerequisite for the instantiation of those connections that are later going to regulate the amygdala. So the working model is that the brain development occurs hierarchically. So given that hierarchical organization, it may be that the early developing amygdala is driving much of the conversation to the prefrontal cortex. So I liken this to the amygdala being the older sister that got there first, teaching the younger sister what she's learned about the world, so that over time, we can get these reciprocal top-down connections from the prefrontal cortex that help regulate over-arousal of the amygdala. Now, with fMRI, it's a correlational measure, so I can't conclude that based on the data I've shown you so far. But now several tracing studies in rodents have confirmed that idea, where amygdala sprouts to prefrontal cortex emerge developmentally earlier than the reciprocal PFC to amygdala. So these data harken back to a 200-year-old quote by William Wordsworth that the child is the father of the man. So I really like that quote because it helps remind me the importance of looking at development when understanding what these circuits mean to each other. So if we simply had an adult vantage point, we often think of the cortex as controlling the subcortex. But if we take a developmental vantage point, we may appreciate that the amygdala is the tutor of the cortex. So to test this idea a little bit more formally, that the nature of connections between prefrontal cortex and amygdala at one point in development are somehow informing the way that the system is going to operate later on, we took all of the participants I just described to you, where we looked at connectivity in response to a stimulus, and we also collected resting state connectivity. So if you're not familiar with resting state, it's simply having your participants lie in the scanner for 6 to 10 minutes, and you perform connectivity analyses, and the result that you get is often interpreted as the functional architecture of the brain when it's not tasked with any particular task. So we can collect both of those modalities at time 1. We let our participants grow up for 2 years, and then we bring them all back and collect those 2 modalities again. Now, age-related changes in connectivity between the amygdala and prefrontal cortex by both modalities land on the same region of prefrontal cortex. And what we find is the best predictor of this future functional architecture, a functional skeleton of the brain, of amygdala and PFC, is the stimulus elicited connectivity that occurred 2 years prior, over and above its earlier resting state connectivity. So that is to say that these data are suggesting that the way that amygdala and prefrontal cortex are co-activated by emotional events in the environment are very good predictors of the way that the system is going to settle into a resting state in the future. So it's this sort of systems level Hebbian type of molding that we're thinking about. And in fact, the strength of that prediction is strongest at youngest ages. So what I'm plotting here on the y-axis are those prediction weights from this arrow. And we see that that prediction is strongest in 7-year-olds and attenuates at points thereafter, because now the arrows start becoming bidirectional. So that finding is consistent with the idea that during childhood we have this more plastic, malleable system that transitions into a more stable system. So these data have really poised us to ask the question formally, is this middle childhood period a sensitive period for the development of these connections? So we've been very compelled by animal studies that have shown that learning that occurs during the juvenile period is enduring and can be used, those stimuli can be used later on in adulthood, often to reduce anxiety. So to demonstrate what I'm talking about, I'm going to present some work of my colleague Takao Hench, who's at Harvard. If you're not familiar with this study, I think it's pretty interesting. If you take adult mice and you place them in an open field, as you know, they will often run to the corners, right? They're like middle schoolers at their first dance. They run to the walls. And one nest could be silent, and one nest plays music. And the naive adult mouse will overwhelmingly run to the silent nest, unless you pre-expose them during the juvenile period to music. And then they will start showing a gravitational pull towards the music nest. And it's specific to a type of music. So if you pre-expose them in the juvenile period to Beethoven Symphony No. 1, then in adulthood they prefer Beethoven Symphony No. 1 and not jazz, and vice versa. Not only is this preference dependent on this sensitive period timing, but if you play them their childhood music when they're adults, they'll start to show a little more brave behavior and wander out to the middle of the field. And that's what's shown here. So in other words, the childhood music or juvenile music played in adulthood will increase the number of center crossings. He's shown that this effect can be recapitulated, reopening the sensitive period in adulthood with valproic acid, and you get the same effect. What's leading to this reduction in anxiety is, these are CFOS data in the medial prefrontal cortex in adults as they're listening to their childhood music. So childhood music, juvenile music is increasing prefrontal cortex activity and reducing anxiety. So we were very excited by this finding for many reasons. The most important one is that you can appreciate, given how long humans take to grow up, identifying sensitive periods is a real task, because if we brought in a bunch of adults and we asked you to tell me a stimulus that you were exposed to only during childhood and not at many points thereafter, most of us would be really bad at that. But maybe music gives us an entree into this question. Maybe not Beethoven, but maybe if we use data from the Billboard charts and we use pop music, maybe we can, with a little more confidence, get at that window when you are age 7. So, for example, let's say we designed this study in 2012. We've really got to get this paper published. Study designed in 2012, young adults come in. We want a stimulus from when they were age 7. So we do the math, and that means we need a stimulus from 1997. So we dug through the archives, and we found a stimulus that we could use. Oh my God. So we'll use the Backstreet Boys, right? Okay, so what we did is we didn't use an open field. We used a different stressor for humans. We gave them, these were UCLA students, and we gave them really hard SAT math, and we stood over their shoulders and watched them, and we told them that they were doing pretty well, but a little bit lower than the average UCLA student, so they should try and improve their performance. And then we gave them rest periods, and during those rest periods, we let them listen to one of 2 radio stations. They could choose. One radio station would play songs from their childhood, so this is Quit Playing Games with My Heart. The other radio station would play songs from their late adolescence, right? So this is Justin Bieber and Ludacris. And the question is, under duress, do they start showing a gravitational pull towards one radio station? So what we found is with increasing stress bouts, people growing up in the US started to show this relative preference for the childhood music. If I break that up into people who reported a lot of listening to music when they were children, that relative preference becomes an absolute preference. Now people are really tuning into the Backstreet Boys under stress. The control group were people who grew up outside of the US and reported not having listened to this music. They're picking instead Justin Bieber because they at least know who Justin Bieber is. However, it was only the people who got to listen to the childhood stimulus who also showed significant decreases in autonomic arousal measured by hand sweat. And the more that people chose the childhood music, the calmer they reported feeling subjectively. And during scanning, it was the childhood music that selectively increased medial prefrontal cortex activity and strengthened that inverse connectivity, that regulatory pattern with amygdala. And the more that medial PFC activated to your childhood music, the less anxiety people reported during the stressor. So this was a very fun study for us to do, but it was an important one because it started to give us more confidence that there's something about this childhood period where the environment can leave an indelible mark. And of course, we're not interested really in what the Backstreet Boys do to your brain. We're interested in a more species-typical stimulus coincident with this period, which is the caregiver. So humans, like many other mammals and birds and some lizards, spend time with their parents. In the human, parents can be the mother, can be the father, can be an adoptive parent, grandparents, and so on. Because this is a learned attachment relationship, right? Nobody's born attached to their parent. You learn that over the first year of life. But what I do want to point out about humans is that we spend a lot more time with our parents than any other species, right? And most species, you have a puppy, it's on the order of months, and then all of a sudden they're independent. And in humans, it's on the order of years or decades, depending on your family structure. So that is an interesting design for Mother Nature to give us because it's a huge energy suck to raise another human being. Right? I don't know what the number is, but someone counted how many calories it takes to raise another human being, and it's enormous. So there's got to be a big payoff evolutionarily for this design. And the idea that we find very compelling is that this buys us as humans this incredibly protracted period of brain plasticity or of immaturity, where we can learn about the massive number of things that we have to learn about. So in other words, parents allow us this kind of scaffolding for our central nervous system and allows us to learn so that when the scaffolding comes down, the nature of central nervous system functioning is reflecting the scaffolding that happened during development. Now, the rodent literature has really produced some beautiful studies showing this intimate relationship between the parent and amygdala development. And I'm going to be talking about rats here, so the parent is always the mother. This is a summary of data from Regina Sullivan's work at NYU, and if you're not familiar with it, in brief, what she finds is the amygdala right after birth is pretty quiet. It doesn't respond to male odor, intruder odor, tail shock, nothing. In adolescence and adulthood, of course it does. There's this period in between where the functioning of the amygdala is entirely dependent on whether the mother is in the nest with the pup or not. When the mother is in the nest, she keeps amygdala quiet through a series of neurohormonal cascading events. So the amygdala of this animal acts like a newborn. When you take the mother out of the nest, all of those processes reverse. So now the amygdala of this same animal acts like an adult's. So the mother's presence is turning on and off the amygdala. Behaviorally, the way this plays out, I think, is very interesting. So, of course, the workhorse of amygdala learning is Pavlovian fear conditioning. So if what her lab has done many times now is take a peppermint odor and pair it with a foot shock and places the peppermint odor at one arm of a Y maze and control on the other. And what this rat pup will do, just like you and I would, if its mother was not there during conditioning, is it would avoid that peppermint odor, right? It does not like the peppermint odor. And that's all amygdala mediated. But if this rat pup learns that association with the mother present, now it will show a bizarre behavior. It will approach the peppermint odor. And that's what's shown here. With the mom, you see this preference for that peppermint odor. So why is that happening? Well, at the biological level, what happens is the mother keeps cort low, which is the prerequisite for amygdala activation at this point in life. If amygdala is not engaging during fear learning, it enables competitive learning systems like locus coeruleus and olfactory bulb to mediate learning. And if they're mediating learning, it produces a preference. At the phenomenological level, why would Mother Nature ever design a system like this? And the idea is that for a young animal, traditional survival strategies are not useful. Fight and flight are not useful if you can't do either one. The survival strategy of the young altricial animal is attachment. So the idea is that regardless of the quality of care I'm getting as a young infant, it behooves me to prefer things associated with my caregiver, whether they're pleasant or whether they're aversive, as in the case I'm describing here. So attachment can trump fear learning early in life for altricial animals. And this may provide a mechanistic explanation for what's commonly observed in young children, even in the context of maltreatment. They will still form a very strong attachment to their parents. It may not be a healthy attachment, but it's a very strong attachment. So the question is, do we see similar learning mechanisms and biological processes in humans? So these are data from Megan Gunner's lab, and what she's shown is in response to preparing for a public speech, which is a wonderful way to activate the stress hormone cortisol. Young children, if they're seated next to a stranger, will do what you and I would do, and they'll mount a beautiful cortisol response. But if they're seated next to their parents, the parent blocks that response. Now, in slightly older individuals during adolescence, we don't see this effect. The adolescents will mount a beautiful court response regardless of the person they're sitting next to. So we collaborated with Regina Sullivan's lab to use her rodent model and design a paradigm we could use with young children. So we presented young children with a blue square that co-terminated with not a shock, but an aversive noise. So it's like loud nails down a chalkboard. It's very aversive. And the purple triangle was paired with nothing. So children learn these associations seated next to either an experimenter. This is just to say they don't like the noise either. Okay, so they learn next to an experimenter or seated next to their parent. They're not interacting with the parent. They're wearing headphones. The parent's filling out questionnaires, but they're present. After this conditioning, the parent goes away, and we put children into a human Y maze. So this is a play tent where children enter the tent, and we place the CS plus here and the CS minus there. And we ask children to go in and pick a door to retrieve a prize. And we show them that it's the same bucket of prizes behind both doors. We just want to know which door they want to get their prize from. Okay. There's a lot of data on here, and I don't mean for you to swallow it all. But what I want to, the point I want to make is that these are acquisition data. So based on a reaction time measure and accuracy, heart rate, and skin conductance, young children can discriminate CS pluses from CS minuses just fine. There is no difference, however, between alone conditions in gray and parent conditions in blue. So parents don't seem to be doing anything to behavior during acquisition. Here's a video of that play tent just to show you what it looks like.
Two signs.
This child is going to come into the tent.
Do you look at both?
That's not an injury, that's how we collect skin responses.
Okay, go ahead.
Okay, so she picks this door and she goes to get her prize. Okay, so when children are conditioned alone, they do more or less what you and I would do, and they're biased towards avoiding that CS plus. But when children are conditioned with their parents seated there, they now show a significant bias towards preferring that CS plus. You may not be impressed with these differences here because they're not as large as the rat data, but I do want to remind you that these are children crawling around in play tents, so I was pretty excited that we saw this effect at all. The other thing that I want to point out is this is a within subjects design. So these are the same children whose learning is switching at the point of recall as a function of the conditioning condition. And that's just to show you it's true in 3, 4, and 5-year-olds. So we also had an opportunity to scan children while they were viewing pictures of their parents and contrasted that with when they were viewing pictures of somebody else's parent. And children are showing now a decrease in amygdala response when viewing pictures of their parents. So remember, I started off the talk saying children have a really strong amygdala response. And now I'm showing that they're showing this decreased response in response to their parents, which we're currently interpreting as parental buffering or dampening of the amygdala, perhaps analogous to what we see in the rodents. In adolescence, we don't see parents really moving the dial that much. The amygdala is doing the same thing whether they're looking at pictures of their parents or strangers. And that's not to say that parents aren't important during adolescence. They absolutely are. It may just be that at the level of the amygdala, parents have started to lose some of their impact at that low level. And pictures of the parents are also strengthening that anticorrelation between amygdala and prefrontal cortex, which we don't see in adolescence again. So the narrative interpretation is that without the parents, children show this childlike bottom-up connectivity pattern, but phasically with the parents, they're able to instantiate this more regulated phenotype in their children. It's also the case that this effect of the parent is moderated by attachment security. So the more secure the attachment relationship, the stronger that effect of the parent. It's also the case that parents are really, really good at potentiating this system, right? So the data that I've discussed so far have been with parents who are very regulated. Either it's an anesthetized rat who's very regulated, or it's a parent who's filling out questionnaires. We also wanted to know, what if parents are expressing fear or distress themselves? So now we fear condition the parents and videotape them, and then we show those videotapes to their children. So this is me getting fear conditioned here to this circle, and then we show that video to my daughter, and we show her the video of somebody else's parent getting conditioned to the square. And the question is, does she learn better from watching my videos than somebody else's? And the answer is yes. So down here means you're learning better. You are disliking that stimulus more. Children learn to dislike the CS plus that their parent conditioned to over and above watching somebody else. And when my daughter looks at this cue, her amygdala will activate more to it than when she looks at this CS plus. So taken together, we've posited that at least during childhood, parents are very good at dampening as well as potentiating amygdala and fear-related behaviors. So we think of parents as this kind of privileged information source to this limbic circuitry during development. And we have some ideas about the neurohormonal cascades that are switched on and off by parents' availability. And the idea is that this sensitive period closes at some point, and the functioning that we see in adolescence and adulthood is reflecting the learning that happened here on an everyday basis. So for the last half of my talk, I'm going to now ask the question of what are the implications of these data for children who experience unusual early caregiving events. So children who experience maltreatment or neglect are experiencing a very different type of caregiving than I've described thus far. So over the past 15 years, I have been very lucky to work with families that have adopted children from institutional care. So children who've experienced early institutional care, which we commonly call orphanages abroad. So this is an extreme case of neglect, because even in the best case scenario, even with the best of intentions, this is a suboptimal caregiving environment. Siri is really into my talk, sorry. So the caregiving is done by staff members who rotate shifts and go home. The caregiver to child ratio is very poor, and so on. So this is a major stressor for a human infant. And there are many developmental risks for children who experience this type of care, but for children that we've seen who've been adopted, there's tremendous rebound across a number of domains, which perhaps isn't entirely surprising because the B environment, the families that adopt internationally, tend to be, or at least we infer, very invested in caregiving, have a great desire to provide care because it's not easy to adopt internationally. And so this is an unusual situation where in humans, where environment A is so drastically different from environment B, and we have a sharp cutoff. We have a sharp distinction here. There's tremendous heterogeneity in outcome. So some children are thriving by every measure, and some children are struggling. And when children struggle, more often than not, the domain that we hear about is emotion regulation difficulties. So the data that I'm going to show you include participants who were largely placed in institutional care at birth or up to 6 months of age, and then the majority were adopted by their second birthday. So this is really pre-explicit memory formation for most of our participants. Now, if we consider this experience a stressor, we have a massive literature spanning decades on stress research, stress findings on the amygdala. So this is chronic immobilization stress in male rats. So different stressor, different species, different age, and different, all male. But there are some important principles here. So the amygdala at a cellular level tends to show hypertrophy and hyperreactivity following chronic immobilization stress, which is a stark contrast to what's seen in neighboring hippocampus, which tends to show decreased dendritic complexity and less function. I think these data are important because what they suggest to us is that stress doesn't have a uniform effect on the brain. It doesn't have this overall dampening effect, but instead, stress is instructing the brain on how to better respond to future stressors, right? So if you experience stress at one point, it benefits you to have an amygdala that's more reactive. And we also see evidence that that same type of stressor leads to changes in medial prefrontal cortex in adulthood. So those are all adult data, but when we look at the developmental literature, I think a much more interesting story emerges. So many findings in rodents now have shown that early life stress, which is often operationalized as maternal separation, early life stress is associated with accelerated development of these stress systems. And I have listed up here a number of findings evidencing that acceleration. But when we first encountered this literature, we were surprised because in a way it's counterintuitive. There's a kind of this heuristic in the literature that stress leads to a deficit in function. But these data during development are suggesting that perhaps, at least during development, there is a slight facilitation of function. And so the idea from across many papers is that this acceleration is an example of a developmental adaptation that the brain makes. It's an activity-based change because the circuitry is being activated by the stressors in the environment that helps the individual meet immediate needs. And if we view these differences in brain and behavior as adaptations, it allows us then to ask questions about the cost-benefit trade-offs. So we have identified in two separate cohorts, one in New York and one in Los Angeles, that following this type of early experience, children are showing hyperactivity of the amygdala in response to things like fear faces. And amygdala reactivity is tightly coupled with eye contact in humans. So we collected eye contact using eye tracking, gives us a high precision measure of ocular motor behavior. But for increased ecological validity, we also measured eye contact during a live dyadic interaction. And what we observed by both measures is that PI is previous institutional care. PI experiences are followed by decreases in eye contact. And the stronger the amygdala response, the higher the amygdala response, the lower the eye contact during both measures. And in fact, the link between early caregiving experiences and eye contact was mediated by this amygdala reactivity. So if we ask parents to tell us about their children's behaviors, we can look at internalizing and externalizing. So internalizing problems are things like anxiety and depression. Externalizing, as the name implies, are behaviors that include ADHD, conduct problems, oppositional behaviors, and so on. And we see increased scores for both internalizing and externalizing following early institutional care. And this is the Cohen's D right here, which is considered a very large effect size. But what I also want to show you is these are the individual participants. This is 373 participants. And you can see that there's a lot of individual differences. And so much of our work now is trying to better understand what differentiates a child here from a child down here, despite the fact that both children experienced institutional care followed by adoption. So these are connectivity data, like I started the talk with, showing connectivity between the amygdala and prefrontal cortex. And we get group by age interactions here. So these are the data I started the talk off with, where children following typical caregiving experiences show a childlike, non-regulatory connectivity pattern, and that switches to that anticorrelated pattern in adolescence. We've replicated that finding in an independent cohort. But following early institutional care, children don't show a childlike phenotype. Instead, their pattern much more closely resembles adolescents and adults. So that's consistent with an accelerated phenotype, but much more work needs to be done there. So the idea is that during childhood, we get this group difference because following early institutional care, we see a pattern that looks much more like adults. However, as I'll explain, we interpret this, or we think that this is an adaptation of the system. So if this is age here on the x-axis, we think that this strong amygdala reactivity induced by early life stress may lead to an earlier onset of adult-like connections with medial prefrontal cortex through activity-dependent processes. But that may also result in a reduction in this putative sensitive period, or reduction in developmental plasticity. So here are data on anxiety following early institutional care. We see that children on average have elevated anxiety. But if children following early institutional care show that adaptation to the adult-like phenotype, then they are going to have lower anxiety than their previously institutional peers who don't show that adaptation. So the idea is that the current anxiety today is benefited by having an age atypical connectivity pattern. That's current anxiety. I wanted to present here today some preliminary data that looks across a 5-year period. The numbers are still small, so I present them with that caveat. But these are the data I just showed you where previous institutional care with the childlike pattern has higher anxiety than those with the adaptation. But 2 years later, and again 2 years after that, that difference flips. So now the children who were initially benefiting from the adult-like connectivity pattern seem to stay with their higher anxiety in the long term. So these data are consistent with the idea that there may be a longer-term cost. And one idea that we're thinking about is that the children here who display more anxiety, and in this case it's separation anxiety, may be eliciting different behaviors from their caregivers as well, which is feeding into that developmental plasticity during childhood. So this is the last set of data I'll show you. These are the amygdala responses to parents minus stranger that I showed you earlier. Following typical caregiving, children show that decrease, that buffering in amygdala response to parents. At the group level, following early institutional care, we don't see that same amygdala buffering. But what you can pay attention to are these very large error bars, which suggests that some children following early institutional care are showing amygdala buffering and some aren't. So we can interrogate those individual differences to see if they have meaning in the long term. So we split groups up into those who showed buffering of the amygdala at time 1 and those who didn't, and followed them over this 2-year period. And we find that in the post-institutionalized group, although everyone had elevated anxiety, if children exhibited amygdala buffering to their parent at time 1, then they showed significant decreases in amygdala reactivity, in anxiety over that 2-year period. And so what differentiates those two groups of children who buffered and didn't? It was not age of adoption. It wasn't factors related to the early history as far as we could gather, but instead, children who exhibited amygdala buffering to their parent also reported more security with their parent. So it was a stronger relationship that they reported with their parent. And that's very consistent with things we've been finding in the lab where this is the children's reported attachment security on the x-axis and internalizing problems on the y-axis, so anxiety and depression. And following post-institutional care, the stronger the attachment security, the lower the anxiety that children are reported as having. So today I presented data showing that human amygdala medial PFC development occurs very slowly, but that there's value in this slow cooking. And the transition to adult-like phenotypes seems to emerge at the beginning of adolescence. This long period of plasticity would then allow for environmental scaffolding, massive environmental scaffolding, particularly by the parent who is providing major inputs into the circuitry that should have long-lasting consequences for emotional behavior. Early caregiving adversity does significantly increase the risk for poor mental health, but there are large individual differences. And it seems that, I think this is the most important message, that strong parent relationships can have really ameliorative effects, particularly following early adversity. And early caregiving adversity may exert its effects on brain and behavior through pacing, so considering the temporal dynamics of development as it's affected by early experiences that we have. And so we think that understanding brain development through the lens of adaptation is a powerful way to better get at mechanisms of construction of this system across 2 decades of life. So with that, I will thank the funding sources, lab members past and present, and especially the families and individuals that contributed their time. And thank you for your attention.
That was a really great talk. So one of the things that popped into my mind listening to you was with the neurodevelopmental hypothesis of schizophrenia, which I'm sure you know about, but for the rest of the audience, the idea is, why does schizophrenia largely develop in late adolescence, early adulthood? And the neurodevelopmental hypothesis basically maintains, well, there are things that are going on earlier, neural problems, developmental problems, interacting, but they don't manifest themselves as schizophrenia until, the time when that prefrontal cortex dampening of amygdala function comes on. And so a lot of times in that literature, I don't necessarily see people referring to, your findings or related findings, but it seems to me like a red light went off, like ding, 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 ding. That's one of those mysterious prefrontal functions that comes online in adulthood that could be a factor. I mean, and not only in schizophrenia, but in other pathologies too. What do you think about that?
Yeah, that's a great comparison. So I think that there are many examples of behaviors, particularly around psychopathology, where they're much more informed by thinking about the hierarchical nature of brain development. So it's the hierarchical nature of brain development, which goes in this kind of backward C-shaped way, where things low and back develop early, and then things high and front develop late. And you guys all know the age at which prefrontal cortex reaches adulthood, right? 25,
Yeah,
which
exactly.
the car rental companies knew before neuroscientists. And so I think that appreciating that hierarchical nature, interacting with what our changing developmental tasks are. So the tasks that we have across development are also hierarchically organized, right? So there's the argument that, well, once PFC, ding, 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 comes on, then you start to see behaviors associated with schizophrenia. But it's also that the tasks, that the expectations of our behavior are also changing across time. So I think about that a lot with anxiety. So I started the talk off saying, well, anxiety, you tend to see this peak age of diagnoses around age 11. That's true across many people. But it's also the case that the developmental tasks are changing, because before that point, anxiety behaviors are even higher in everybody, right? So you think about the average 6-year-old or 4-year-old, they're scared of the dark, witches, under the bed, being apart from mom, strangers, clowns, dogs, everything, right? And so those are all normative behaviors, and they decline with increasing age. At around 11, some people aren't declining as much. So you start to see that separation, and teachers are starting to notice, and parents are starting to be able to compare their kids' behaviors to others. So I think it's both the hierarchical nature of the brain as well as the environmental tasks that we're put to.
Another question right here.
Do you think your findings from the pre-institutionalized kids could be extended maybe to a lesser degree to kids whose parents were forced to, for whatever reason, put their kids in daycare at maybe 3 months of age?
Yeah. So thank you for your question. So the quick answer is no, but I'll unpack that. So did you all hear the question? So the reason why we wouldn't expect those data to translate to things like daycare, or and the reason why it's been shown that those data don't translate, is because there's a fundamental difference between what children experience in institutional care and what children experience when they go to daycare, which is, in daycare, there's a rhythm that you're always returning, right? When my kids were in preschool, actually, my daughter's here, when she was in preschool, there was a mantra, mommy always comes back, right? And you learn that patterning day in and day out. With institutional care, there is no rhythm at all, and it's 24 hours a day without a parent. And even if you have staff that are taking care of you, they could get fired at any time, right? So there's no stability in the caregiving, which is the important input for developing these attachment-related systems. What we are interested in doing is, now that we have these data, we have a new project that's asking how much of these findings with this very extreme environment, very extreme deviation in the environment, how much of these findings apply to children who experience other forms of early stress? So children who experience less extreme but more normative forms of neglect, even in the home, children who experience other forms of maltreatment, what can we expect to be similar and where are there differences?
I think a lot of us parents are probably singing Daniel Tiger now with the grown-ups always come back, but that was amazing. And I really loved how you would go back and forth from human to rodent research and you brought it all together. I was really struck by the work that you had done with Regina's group. Oh, I'm sorry. Stan? Okay. I was struck by the work that you had done with Regina's group and kind of blown away by the animals and people aren't just learning more about something, but they're learning the opposite. And we used to wonder that when I was in graduate school, you'd see moms stepping on the babies and biting the babies. And we always wondered, why is it that they don't form an aversion to that experience? So there's something interesting that happens when they're with mom. And here, what you're showing is that they're not just learning about it to a greater degree. They're specifically learning that it's not aversive, but in fact, it's pleasant because they're showing you a preference for that later on. So I find that really interesting that if mom is there and if mom doesn't react to the experience, this bad thing isn't really bad.
Yeah.
So what do you suppose would happen if you were to combine that with the next set of data that you showed us where you're showing your children images of their parent experiencing fear conditioning? So what if now instead child is experiencing an aversive conditioning experience and mom is too? In that case, might you expect the children to have an augmented response in exactly the opposite direction?
Yeah, it's a great question, and you made me want to say 10 things. I'm going to try and give the short answer. So the prediction is spot on, that the children should learn themselves even better if parents have a very large reaction. And there's so many examples from neuroscience that we could turn to. There's a huge literature in dental fear that we can turn to, right? Thinking about how parents scaffold that experience for their children. Thinking about math anxiety is a new area where people are looking at what are parents' reactions to children's math anxiety. There are examples back from World War II where in London, following the air raids, children's shell shock, what they called back then, was often a function of their parents' reaction. So this is not to blame the parents at all. I mean, I think it's actually to empower parents, that parents are really a conduit, an interpreter of emotional environments. So I think you're right that it is fascinating to think that what they're learning is actually that, you know, this is okay. And actually it's sort of like that Proust story of the smell of the cookies from when they were young. It's like bringing me back home to smell that. So I think about, like, you know, my father smoked cigars, right? And I know people hate cigars, but there's something compelling about it, right? And so I think we all have that experience where that set of stimuli means the nest somehow.
We have a student question here.
Hello. Sorry, I'm a nervous wreck. That was a really interesting talk, as I'm an undergrad student and I feel like the baby of the neuroscience field, like just now learning stuff as I'm only like a second year. What I found really interesting was the connection with the prefrontal cortex and the amygdala, and how I associated that with like dissociative identity disorder, and how when they develop based on the neglect they had as a child, like those emotions and the connection to the prefrontal cortex. Would you like use your research to go off of that? Like, could that be used for that?
Yeah, well, thank you for your question. So I haven't ever studied dissociative identity disorder, but I think that if we start with these fundamental questions, like I've oversimplified a lot, right, just to start to build a model. But I think that with that model, we can then ask about much more complex behaviors like personality disorders, right, where that's the entire brain to, you know, for our personality. So I think it's a great question, and that's the way we would start to go, is how does the cortex start to construct meaning of who we are? I think that a lot of these early bottom-up processes are going to feed into that.
Okay, sure. I'm actually going to let you ask your question afterwards. Let's go. Okay. Are you a student? You're a faculty. Sorry. Do we have another student who has a question?
Hello. So you are talking about the aversive parental reactions and how they are sort of work as a child to the child's benefit, whereas we would see them as aversive. The child is learning. Sorry, I'm trying to collect the question. Anyway, so what you're talking about, your further studies are about child neglect and maybe maltreatment. Would you anticipate that child neglect would result in more maybe like a maybe not as like a developed, like not on time, like maybe a delayed amygdala connection with the prefrontal cortex, maybe because there's not a parental figure or caregiver that's actively enforcing the fear response or actively teaching the child in a way. So since there's an absence of that, would you expect that there's like a delay in the circuitry?
Yeah, thank you for your question. So our data here would point me in the opposite direction, that there is an acceleration of development. And the hypothesis is based on the idea that if you are receiving cues early in life, that you're on your own, that there isn't a sensitive environment around you that's going to provide security and safety, then it behooves me to switch strategies and not, bask in the wonders of childhood, the luxury of childhood, but I need to get these gears turning at an earlier age. And you see that behaviorally in the clinical literature where children who have parents that aren't able to provide good parenting, maybe they have their own mental health problems, maybe they have severe addictions and they can't parent, children often show a little bit of growing up quickly, right? This early parentification behaviors, starting to engage in adult-like behaviors at earlier ages. Maybe that one way to think about that is that's kind of growing up quickly a little bit. And that's adaptive in many ways, right? If I am a young kid and I can feed my siblings breakfast in the morning, that's adaptive. But maybe there's also a cost we want to be paying attention to in individuals who seem to be growing up quickly.
Okay, so I want to thank Dr. Tottenham for a wonderful talk and for great questions. If you have more questions, she'll be available afterwards if you want to ask her some things.